Namaskar to our audience for our next talk on Karmayogi Bharat. If all of you remember, we'll have one talk a week for the next 52 weeks on a set of subjects that are of interest to a large number of people. Today's topic is a very, very interesting and useful one as we look to the future. One of the technologies that will change the world is genomics. And we want to make sure that people have a good view of the what, who, and why it matters of genomics. And we are very grateful and privileged to have a real expert on this who has done groundbreaking work in India on genomics with us today. So Professor Mitali Mukerji is currently at IIT Jodhpur. She's a distinguished professor in the Department of Bioscience and Bioengineering. She is affiliated with the School of Artificial Intelligence and Data Science. And before joining IIT Jodhpur, she served as the chief scientist at the CSIR Institute of Genomics and Integrative Biology in Delhi, where she also led the Department of Genomics and Molecular Medicine. Her pioneering research focuses on understanding the human genome structure and variation to discover biomarkers for disease susceptibility, human phenotypes, and environmental adaptations. She has been instrumental in initiating groundbreaking genomics projects in India, including work on rare genetic disorders, population genomics, functional genomics of repetitive sequences, and Genomics, an innovative field integrating genomics with traditional medicine. As the convener of the Indian Genomic Variation Consortium, she contributed to creating the first comprehensive genetic landscape of Indian populations, with insights influencing studies on human migration, disease history, and genome-based diagnostics. I won't go to all the other things she has done because it's a very long list, and we are all very keen to hear from her as to, what is this technology that is that is changing the world and will change our future over the next 20, 30 years in many ways, including in medicine, in our understanding of history, etc. So without further ado, I will pass it on to Professor Mitali, who has a wonderful presentation, and then we will take questions from the audience at the end of that. Professor Mitali, on to you. Go ahead, Professor.
To speak to this audience in this portal. It's wonderful, and I love this subject, and I would like to maybe pass on some of the excitements over the next 45 minutes or so. So I'll start sharing my screen with your permission.
Yeah. Please go ahead.
Yeah. So are you able to see my screen? Yes? Hello? You're able to see my screen? Can somebody confirm?
Not yet, ma'am.
Not yet?
We can see various boxes, but we are not able to see your presentation.
Uh-oh. Let me try this. Okay. Are you able to see now?
Yes, you've done it. It's there.
Okay. So today I'm going to talk about the science of genomics. As is mentioned, what is it? What do we gain out of it? And over the after the lecture, if some of you have questions, then I'm also ready to be there for addressing them. So first, let me try to put things in some kind of perspective. That, as we all know, that at this point of time, everybody is looking for solutions which would tackle all the aspects which bothers us comprehensively. And this is a framework what people are now calling as One Health framework, where we want to ensure the human, the environmental, and the animal health are all in sync, and to be able to tackle the more and more emerging problems of today. For example, when COVID struck us, we all got to know that it was a zoonotic spillover because the habitats are changing, and then it spread over all parts of the world. And many things came to fore, where we got to know that we need a multitude of people participating together to tackle things comprehensively. And these are in spaces of trying to uh, cope up with climate change, the pollution, the biodiversity degradation, the spread of pathogens, the spread of antimicrobial resistance, ensuring food security, the health vis-a-vis our diet and lifestyle, and also all the other kinds of social determinants which impact our health. And all of these need contextual solutions, which have to be affordable, it has to be scalable, it has to be precise, and if we are able to do it in a early interventions, then these will also be much more affordable. And more importantly, in a country like us, where it's extremely diverse with large number of people, it needs to cater to the diversity and also the context. And above all, whatever solutions we want to evolve, it has to be sustainable. So at IIT Jodhpur, we have initiated something in the One Health perspective, uh, in the context of Thar, which is the desert ecoregion. It's one of the most populated deserts of the world, with a large number of constraining conditions of heat, aridity, scarcity of vegetation, and people have adapted over here, but they are at the ebb, right? So if any climate change happens, it's going to impact a large number of people. And how do we evolve a comprehensive way of looking at the entire Thar is what we are doing in this program of Thar Designs. The other is IoTech, which I'll come to a little later, which also is how you can use technologies to build bridges between our modern medicine and traditional medicinal systems, to be able to provide more options to people by way of managing their health. So, why this is pertinent at this point of time is because people all over the world are talking about precision medicine. That's the only way by which we can actually have uh, affordable, targeted way of managing our health right from pre-womb, that is when the child is in the prenatal stage, to understanding the cause of death, so that preventing death, and across the spectrum, various aspects where you want to prevent the disease, you want to treat the disease most effectively without having most side effects, or you want to increase the health span, what is being called as health longevity, or you want to control infection, spread of pathogens. So this is the entire spectrum where we would like to evolve individualized solutions. And more and more, as AI is impacting every sphere of our life, so also in the health, it's being thought that AI could be a big enabler in this area, where we can now start to think of scientific wellness if we were to capture all our information in spatio-temporal dimension, just like a GIS. So if we are able to collect all the information, and each of us have a big data around us, and where any change we have, it will get reflected in our virtual twin, so that we are able to make suitable calibration and manage our health across the health span. So this is what people are talking about. Today is the age of scientific wellness, but is it achievable? How do you get to it? And the other thing is also, people are now, when we are talking about this management of health in a personalized manner, such a thing also existed and is still contemporarily practiced in our country, and it's also being acknowledged all over the world. However, how does one mainstream Ayurveda when things are written in an entirely different language? The systems, the ontologies are very different. how do you today enable a crosstalk so that if something has been existing for 5,000 years, could we query it and then try to see whether we have more options if we take it? Then the other important aspect is that we see animals all around us, which has also evolved. We tend to think that since we can speak, we are bipedal, we have tools, we can talk, we have been able to control fire. We think that humans are the most evolved. But if we look around us, there are very, very many animals which would be size of which we cannot see by the naked eye, to an elephant. Each of them has a very, very intelligent design in which they are made, and many of them are revealing very interesting attributes. For example, there's this Deinococcus radiodurans, which can resist radiation. Tardigrades, these are called as water bears. It has been to space also. It can remain in an entirely dehydrating condition. Then there's this naked mole rat, which doesn't have cancer, which lives very long, 30 years, where the average lifespan of a rat is maybe a year. Octopus, we know, are solvers. Python can eat an entire animal without compromising its digestive system. It has to expand the entire head and jaws to be able to accommodate a full animal. Elephant lives very, very long, doesn't have cancer, minimally. And then there are fruit flies which are around bananas, and humans have been able to see that their body design and their brain are maybe many, many parts of the nervous system can be recapitulated in Drosophila, or the honeybees, where we know they practice social behavior. During COVID time, we did not know how to kind of work our way through crowded places, but that's honeybees' way of life. So how do they do it? And can we learn from them? Will that learning enable us to also have better ideas? So this is where the genomics comes in. You have to believe me now, but when I go a little later, you can see how introducing this technology of genomics can actually provide answers to many solutions. How does that happen? So I have written some words of some a sentence. We can do it, but it's written in so many different languages, and I'm very sure most of you can read one or two of them, and you cannot read all of them. So that shows that you have the same expression, but if you write it in different languages, maybe you might not be able to make effective conversations, right? But the beauty of biology is all life forms are written in the same language. And this language is the language of DNA, the language of four chemical letters, A, T, C, G. And all life forms from bacteria to plants to humans are all written as to how they have to be designed, are written in these four chemical letters. So you have different expressions, but you have the same language. And this is evident how COVID could infect us, because SARS-CoV-2 could actually talk to the human cells, and that's why it could infect. And that it could do because they have a common language of DNA. Microbes colonize us. How do they live with us? Because of sharing of language. And we've been able to also kind of introduce a gene from human into a bacteria to produce it in a large number or amount by genetic engineering. And that is also possible because these genomes can talk, or there is a cross-talk between the languages. And it might be worthwhile for you to maybe remember, most of you might know, that we have trillions of cells in our body, and each of these cells have 23 pairs of chromosomes. And these are there in all the tissues, all the organs, and the entire organism. In all the cells, all the chromosomes are there. So the amount of information we carry in our body from a cell to an organism level is immense. For example, if you stretch the genetic material in our nucleus, it would be 2 meters. And if you stitch all the genetic material in all the cells, then you might be able to circle the solar system twice, like 70 billion kilometers. But all of that is compacted, and all of that actually opens up when it has to function. So how does that do? That four chemical letters, how does it get into meaning? So I can just give an analogy, just like you have English alphabets. These nucleic acids are to our book, and if you thread them like words, then it's what is called as DNA. And if you thread them again, it becomes sentence, which has punctuation. So you have sentences like which you can read at a time. So these are akin to something. The genes are akin to some sentences, and if you put all the sentences together, just like you have a chapter, you have a chromosome, and if you put all the chromosomes together, it becomes a book, and that's a genome. So we inherit one such book from each of our parents, and that's what our genome is. It's these letters are 3.3 billion in number, immense amount, which if you were to write the letters and read it, you'll actually take 95 years non-stop to read it. But our genomes are doing this reading every time it's making a decision. So how does this happen? How does the genetic material convert to an information which governs how we look like or how we behave, etc.? So what happens is the DNA is a blueprint, and the DNA gets converted into a messenger by a method which is called as transcription. So the DNA, it is a double-stranded. One of the strand conveys a meaning to make an messenger, which is called as mRNA, and these are read in threes to something called codons, which actually governs an amino acid. So what is an amino acid? If you thread all amino acids together, you get a protein. So protein are the functional entities most of the time. So these proteins come from the DNA being read through a messenger. So you can understand that any change in the DNA can impact the RNA and then can impact what kind of amino acid will be there. So the protein might have a different amino acid introduced, and as a result of which, sometimes there could be a difference in the functioning of protein, or sometimes there could be such kind of a striking phenotype. This is an RBC, and this is a sickled RBC. So you would have heard about sickle cell anemia, which impacts a large fraction of our populations in the central part of the India. This is because of one kind of a change in a gene which makes this RBC, and that's what causes sickle cell anemia. This is a rare disease, but such kind of diseases are 6,000 to 7,000 such rare diseases are known. And maybe if you combine all the rare diseases prevalence, it impacts 1% of the population. So estimates are in India, 70 million people would be living with rare disease. The lifetime management of a rare disease could go as high as 10 crores, because it's a very, very difficult disease to treat and manage throughout life. Now, such kind of mutations or variations, as they are called, are rampant, and there are different kinds of variations. So, for example, I start with the same analogy that if you have color, sweat like this, you can have another color which there is a drop of a letter, it's a deletion, but it would convey meaning, so it can remain. But over time, if there is an insertion, then it can have another word which would not have any meaning, so it has to survive, because so a spell check can make it cooler. So that is what happened. A substitution happened, so you have another word. Then there could be another round of deletion, so you could have cool. You could spell it in today's language, and it would become cool with K. And you could also invert it, so it becomes look, or you could have a fusion, so it becomes cool look. So or it could take a different trajectory, and it could actually get into another word which has a meaning, but a meaning very different from where we started. So these are what are changes in the letters or in the DNA, where you can have single change, you can have an insertion, you can have inversion, you can have duplication, as a result of which what you start with and what you end could be very, very different. When do these changes happen? So as the DNA, the cell has to divide in number, it has to replenish itself. So its DNA also has to divide. And during the process of dividing such long pieces of DNA, you could make errors, just like you do it when you are writing a long document. And these errors, if they creep in for at quite a number of places, you can actually have a very striking differences. So these are what are called as polymorphisms. If they are very rare, they're called as mutations, or depending on how frequent they are or how rare they are, based on how many number of people in a population would have those variations, it gets defined as rare variations or common variations. These are the changes which govern diversity. So we are all humans. So at some core places, we are fundamentally similar, but even between our siblings, we differ. So these could be because of these small, small changes. So, in an effort to read the entire book of life, because it was thought in the 80s that if we knew the entire book of life, then we would be able to have a reference with which we could compare every other genome, and we would get to know the basis of differences amongst people, why some people are susceptible, why some people are healthy, why some people can colonize high altitude, etc. We could get to know once we have a reference book. So that was what the Human Genome Project was all about. It went on for 13 years because at the time every technology to read a genome was getting built, and the first draft was announced in 2001. And this was an expensive, extremely expensive enterprise, but this was the beginning of genomics. What is very interesting is that, just like I mentioned, that all of the organisms shares the same letter. So pretty much any kind of organism can be, by the same tools and technologies, can be read. So the genomics has actually federated the entire learning of biology, just because the same letters are shared across animals. So typically what you could do is you can collect a sample from any source, you can isolate the DNA. It's typically in there's a process. Then you can see whether it has the DNA, whether it has RNA. So there are methods to do that. You can actually amplify a small bit to a very, very large amount, so that you can, if the DNA is a very small amount, you can amplify that. And then you can also read it, so that you get to know what other letters. So there are lots of technologies behind that. We'll not get into that. Suffice it to say that now we have the technologies to read it. And this is what happened. So we were very ambitious. We thought that we will get to know at all times. We as humans think we are the most evolved. Earlier times, there were paradoxical things, that if we thought we are evolved because we have more number of chromosomes, that is not so. A grass has more. Then we thought that our genomes might have more sizes, so that we are more complex. It turns out that a single cellular amoeba has more content than the 3 billion Homo sapien. Then there was a betting that when the human genome comes out, we will get to know that we might have more genes, because of which we are more evolved. And it turns out that the small fly and the human has approximately the same number. So what is happening? How do we get to be so complex? What turned out to be very interesting is that there is a huge amount of repetitive sequences, which was earlier called as a selfish DNA or the junk DNA, and the amount of the interspersed repeat has actually increased in comparison to the other. So now, then the thought was that, are we human because we carry a lot of information in a part which was earlier thought as junk? The other information which came up was that the humans are colonized by more than 1.5 times bacteria, and they colonize all parts of our body. Different bacteria reside in different regions depending on what food they get. And as a result of which, if we have trillion cells, we have 1.5x bacteria. So what is human? Humans are nothing but a metagenome with human and bacteria, because these bacteria colonize us right after birth. So these are some of the surprises with which genomics gave us. It was very humbling. And over a period of years, what took us 13 years can be done in 2 to 3 days now, because of extreme unparalleled advancements in sequencing technologies that had increased the number of people we can do at a time, the miniaturization. So now this is a handheld device from what a big device we had, where we had to have ACs, computers. Here, everything you can do it with your mobile phone. You can start the sequencing, you can carry it with you, you can take it to the forest. And it has increased the access. That's why because of the lowering cost, and the ease of doing genomics is also increased. So as a result of which, huge number of genomes after the human genome sequences have been done, and now there must be in the public database some 500,000 genomes of humans also. There are large number of initiatives, because once this people got to know that you can do this, then in a One Health perspective, you can pretty much sequence everything in the local biota, and you can start to see what are the differences which are creeping up, let us say because of climate change or because of some pesticides or etc. And that's how the lots of projects started in UK for sequencing all kinds of birds, all kinds of animals. So there's a Earth Biogenome project, very ambitious, which wants to sequence every living organisms in the world. So these are the things which have been done. But is it really useful? Is the next question we have to ask. We can keep on sequencing genome. What does it yield? So, very interestingly, like I started in the beginning, it is yielding curious answers. How does a python engulf the entire thing? So its body gets heated like a pressure cooker, you can think of. So its mitochondria functions differently during feeding, so that it's able to digest the entire food as a meal. Uh, uh, these naked mole rats have a very efficient repair mechanism so that it doesn't get cancer. It has newer, it has interesting genetic makeup for which allows not to have pain. Again, cancer doesn't strike elephant because it also has copies of genes which are tumor suppressors. So they suppress the tumor. These are the genes which gets mutated in the cancer condition. And then newer, what are the cognitive abilities in an octopus is also getting revealed by some kind of transposable elements which are expressed in the brain. Very, very striking. It has also provided us an information as to how we lost our tail. So during the course of evolution from the primates, the humans or the primate lineages lost their tail. So by genome analysis, one found that there was a defect, or there was a change in the gene because of which the humans lost their tail. And the proof of the concept was, if you take that and put it into a mouse which has otherwise a long tail, the tail is lost in the mouse. And what is very interesting is this is because of a transposable element. The what was called as junk, that junk has contributed to some feature which is very, very unique to the ape lineages. So these are very, very interesting facets which are coming out. Very recently, there's an interesting living fossil which actually breathes by gills also. It's called the lungfish. Its genome has got sequenced, and the most surprising was that it's actually, if the human is 3.2 GB, the size of this genome is 43 GB. What could be happening? What is this doing with so much of amount of DNA? And what is very interesting is the gene numbers are again the same, but the repeat content has increased to 90%. And this is a very, very resilient fish which can live in water, which can live in very dry conditions. So people will mistake it for mud. They could build houses, and if rain falls, then the lungfish could come out of its estivation. So many, many interesting attributes have been given to the genetic makeup of unique genetic makeup of the lungfish, because of which it has got a unique way of walking on the land. And this is thought to be one of the intermediate species when humans or life evolved on Earth from water. Now that was all about the genome. Next, we come to genome differences. What do they tell? What do you do with so much of variability in the genome? Because there could be immense combinatorial possibilities once you know that there are 3 billion bases, and one in every 1 million place there is a change. The change which I mentioned earlier. So it turns out that you can actually use this information in very interesting ways. For example, you can get to know the history of how humans migrated from some place to the other. Fossil evidences are there that we evolved in Africa some 100,000 years, and then after that there have been movements all over the world. They have been able to colonize different parts of the world. So can we see these imprints in today's DNA? So what happens is, the further apart you are in age, as in time, more number of changes will happen. So similar genomes will have more similarity, and dissimilar genomes will have more differences. And you can use various kinds of approaches which are mathematical to be able to look at how distant are two human genomes from each other. So there was a major project where they looked at populations all over the world, from Africa, Middle East, Europe, South Asia. It was called as a Human Genome Diversity Panel. So they collected samples from people all across the world and looked at differences all across the genome and then clustered the information. So here, each line over here is one individual coming from different African population, Middle East population, like that. And each color represents how close they are. So if there is so you can see African populations are distinct from Middle East, but Middle East has a little bit of a green. That means they are admixed with Europeans. Europeans are again more similar to each other, and then they are distinct from South Central Asia. East Central Asia are distinct from them, and then Oceania, and then America. So as people have evolved or moved, the genomes have also started to be dissimilar. And by these kinds of things, you can know which population is more closer to which population. So supposing a disease is there in one genetic makeup in one population, then you can predict that this, if this has moved to another part, then likely that it would have carried the disease genes with them. So such kind of things can actually allow you to track populations, track mutations, and all of it. So in India, when this was being done, we did not have a map. So we started a very big project called the Indian Genome Variation Project. It was a consortium of 200 people. Professor Brahmachari initiated this as a convener of the project, and I got to experience a lot of different populations. We looked all across the country from various linguistic, geographical, and ethnic groups, and we try to see how we are related to each other. Can we tell how many reference Indian populations you need to capture the entire diversity of India? Because we have got huge number of populations, endogamous populations, social structure, marriage patterns, as a result of which this will all impact the genome. Nevertheless, once we analyzed it, we found that Indians could be clustered into 5 major clusters. And this clustering was primarily first on the basis of ethnicity, then on the basis of geography, and after that, there would be subtle differences amongst populations. So we had Indo-Europeans in over here more similar to each other in the north, and this part of the population, which were Dravidian or Austroasiatic populations, but isolated populations, are more similar to each other. Dravidian isolated populations were very different from Dravidian caste populations. And the populations lining the Himalayan belt, whatever language they spoke, were more related to each other. So this is the first time we got a comprehensive genetic landscape of India. And what could we do with this? So it turns out, if we started looking at genes and differences of the genes in this kind of space, we find that there are different patterns. So for example, an X gene, which is involved in resistance to malaria, actually had a different distribution across the country in terms of variation. And this was very interesting that its distribution actually kind of was similar to the malaria endemicity map. So that could tell that if people are residing over here in malaria endemic region, they have a genetic makeup which allows them to survive in malaria. So you could actually discover what are those genes which allow you to survive in malaria, and you can use it for other places where risk for malaria you can predict. Similarly, we got variations which distinguish populations residing at high altitude compared to sea level population. This tells us that what is those genes which allow one to habituate better in high altitude. Maybe it would be useful for example, sojourners who go to high altitude, or army people who are residing in high altitude. Like this, we got a whole set of pattern, and this actually told us that how diverse we are and what was the importance of having such a reference map made. Calcutta Museum actually houses our work, which tells that how this revealed that all of us, how we are different, and we need to kind of appreciate this diversity. We became through this we became a part of a Pan Asia project where we wanted to know how people populated different parts of Asia. It was a very striking work. We 10 countries worked together, 72 populations we looked at from Asia, Southeast Asia, and I led the project from India, so got to again learn about more about population genomics. But it revealed a very interesting insight that earlier people thought that there have been multiple waves of migration into South Asia, but we refuted this. We found that there was one major movement to India, and after that it radiated to different parts of the country. So India has been a major corridor for peopling of Asia, was what came out of this very seminal study, where a large number of people participated from different Asian countries. This These kinds of information becomes relevant or became more relevant when we as we lived through the COVID. As all of you would have seen that the rates of mortality per million people was very different despite population sizes. So many parts of the world, mortality was much higher compared to India, China, Zimbabwe, which actually tells that differences of in individuals, and these differences in individual who make up the population can actually give a different kind of a phenotype to a populations also. And it could actually, there's a very interesting book called Genes, Polymorphism and Making of Societies, which actually delves into how these variations which has shaped the human populations could contribute to the way our outlook in life is. For example, Westerners, why they have such an individualistic living compared to South Asians, where we have a collective society. And many of them, he's been able to very compellingly able to show how the way our society is built has a lot to do with the way we are genetically made up as a population. Of course, there are individual nuances. So those foundational days actually led to a large number of projects across the country, which were to tackle rare diseases, cancer, know the populations of India in a larger scale, and these are all ongoing across the country. And it has actually opened up newer avenues in the healthcare domain. I apologize, I'm not doing a presentation on the plant genomes, but similar kinds of advancements have happened in the plant. Since I don't belong to that area, I would not have done much justice. So I thought that I will talk about the genomics from this perspective. But whatever is to animals or humans would be similar to the plant, agriculture, etc. And because of these kinds of foundational setups we had during the COVID time, we would be able to track you people, everybody got their RT-PCR done, got to know how the strains are evolving. How do you get to know the strains are evolving? You just get to know by sequencing them. So these sequencing technologies, which were used for various other kinds of studies, got reoriented to tackle SARS-CoV-2. And as the mutations were evolving, people could track them in real time, and you could actually make a map from where a mutation has originated and where it moved, and there if the new mutation has originated, everything could be tracked in meaning in the during that ongoing pandemic. And this led to this big consortia called INSACOG, where people all across the country started sequencing, depositing their sequences so that one could actually use it for surveillance. So this was the foundational aspect in our country about large-scale genomic surveillance, which can now be channelized to addressing any other surveillance problem or emerging problems across the country. And this is about variations, but what are the new things genomics is providing? One of the things which genomics definitely is providing is how to use it for forensic application. Just like you have the fingerprint, the DNA of a person is also very unique because there are so many places you can have variations. So the combinatorial leads us everybody to have a unique genome. So if you have that, then any crime site, you can actually take any material, small material, hair, semen, any material, and you can amplify it, get the DNA, sequence it, fingerprint it, and you will be able to uh, uh, use it for different kinds of forensic purposes. The best example was of this one, which was kind of, I find it very interesting. Richard III in UK. When he died and he was buried, people did not know where he had been buried. So somebody found it in their porch. They found there was this coffin, and when they opened it, they thought that it seemed to be Richard the Third's skeletal remains. And then they used a variety of technologies, but the most clinching thing came from analysis of their genome from the because you can take the DNA out of any material which has a nucleus, and they could trace it to a maternal lineage who had an ancestry of Richard III. And then based on the DNA, you could predict the eye color, the hair color, and there was a portrait of King Richard III to which there was a match based on the date, the skeletal structure, the DNA with the current living member, and the what DNA tells about the eye and hair. And they could say that after 500 years, they could locate a missing person using genomic technology. You can use it for very other things also. Like I mentioned, the DNA is universal. Not only is the DNA universal language, but for certain traits, it's also shared across species. For example, there is a gene for color of skin, which actually gives differences in color to tigers, to fishes, to bears, and also to human. And if you have a defect in this gene, it also leads to certain conditions like vitiligo or demyelinating skin colors. So once you understand this, then you might be able to find what gene is defective, and there is a hope to then rectify it. So when so many genomes are getting sequenced, and if you know the curiosity of around peculiar features of any one of these genomes, you have a hope to find out the reason behind certain kinds of rare diseases by just looking at these sequences and comparing it in a mutant condition or in a disease condition. So this is actually called as phylomedicine. That is, you are using an evolutionary approach along with genomics to understand what is the cause of certain kinds of diseases in today's day. You can also look at how people kind of survive in extreme adaptation, which is very important for a navy people or people who go to high altitude, long distance flyers, etc. So there is a sea nomad, very interesting, Bahau people who actually can dive as low as 70 meters and they stop breathing, just like the seal. So people were curious what has given them this advantage, because this could be of very, very important use for deep sea divers. So they found that the spleen size of these people are bigger because of a mutation in a particular gene, and that bigger spleen size allows them to store RBC. So whenever they are diving, it actually can release that so that they are able to cope up with the low oxygen condition. So this is a very, very interesting attribute one can innovate based on this observation. The other thing, I think 2022, many of you have heard a person called Swante Pabo. He got a Nobel Prize in medicine because he did something very phenomenal. He actually was able to provide technologies which could sequence from fossils and recreate who our ancestors were, how were their genomes. So Neanderthal, Denisovan genomes could get sequenced. And in today's day, 2 to 5% of our genomes have a Neanderthal origin. So this was the first time it actually kind of revealed that there would have been some time when Neanderthal and humans would have coexisted, and maybe some of the ancestors could have mated, as a result of which some of the genes from Neanderthal could have come into humans. Some of them would be giving us an advantage because Neanderthal were colonizing extreme habitats even before humans started moving out from Africa. But today's date, a large number of diseases related to skin, malnutrition, hypercoagulation, if one were to look at the DNA which causes that, it is traced to a Neanderthal origin. So, how we have inherited some traits from even predating human comes out of this ancient DNA. It also reveals some very interesting things, that if you know ancient DNA, could you actually do something for today's date? Meaning, why it is important to resurrect the past through DNA. So there was this period of Black Death, a bubonic plague, which went on for 500 years, and nearly 50% of the population in the Middle East, North, and Africa were wiped out because of this bubonic plague. But there were skeletal remains which could be exhumed, the DNA could be taken out. And through this ancient DNA technique, not only were they able to see what was the virus which caused this bubonic plague, called Yersinia, but they were also able to, because the dating of the in the cemeteries were very good, so they could take the remains of people right after plague and before plague, and looked at what was the genetic makeup of those people who were susceptible to plague. And very interestingly, it came out that the people who would have survived would have a very highly evolved immune function, because of which they could survive through the plague time. Today's date, those variations still exist. And these are the ones which are also associated with a lot of autoimmune conditions, because our immune system is on an overdrive when you have an autoimmune condition. It is because of the past adaptation to a very plague-like condition, which made us gear up for tomorrow's pandemic like condition. But it also now, when we don't have any such kind of microbes around us, then it could turn around itself, and we would have the conditions as we have today. Then there is another interesting, so this is all about how you can get from the past something which you can now use this information in today's time. But there are other kinds of things which you can also do. So there is another area of something called metagenomics, where you don't know what are the organisms in a particular ecosystem, but if the organisms are there, their DNA is there. So you can actually do a full community DNA analysis. And then from that community, when you have the sequence, you can identify what would this community belong to, what kind of microbes would have been there, what kind of animals would be residing. Not looking at the animals, but by looking at the community DNA from a scat sample, from a gut stool sample, from a leech's blood. You can pretty much take out DNA from everywhere. And if and looking at the DNA, you can know whether the leech has sucked on whose blood, or in your gut, what kind of microbes are there. So this is the area called metagenomics. And people have used it in a very creative way for biodiversity monitoring. For example, leech can suck the blood of animals, and the blood can stay for 8 months also. You can take out the blood, sequence it, and find what animals it would have fed on. And by that, they could also, in Indonesian forest, they could find some animals which they thought had disappeared, but they are there by looking at the DNA from the blood, because it had sucked the blood from that animal. So similarly, people who have obesity or who are thin, they have body microbes which are very different, and gut metagenomics actually is revealing why people have different kinds of phenotypes. You can also look at by the DNA as to how this could actually contribute to differences in why some animals are living and why some animals are getting extinct. So if you have reduced diversity, then that reduction in diversity does not allow you to cope up with some changing condition, because the more diverse you are, more kind of variations allow you to cope up with different kinds of condition. Seems that animals which are on the verge of extinction, at many places, there is a loss of this diversity, because they don't have enough kind of diverse mates to pair with. right? Or if the habitat are getting degraded, then they don't have enough numbers, their numbers dwindle, then their genomes will also start to get extinct. So you can get these signatures by looking at genomic data, and then you can start to predict which animal is going to go into extinction, so that you can actually have much better way of conserving the biodiversity around that region. Or you can also do some kind of a captive breeding, where you can get to increase the diversity and make the numbers make it not, meaning allow it not to go extinct. So such a kind of a thing has been done by this person called Stefan Schuster. He calls this area as museomics. He looks at museum specimens all across the world, finds out how the mammoth was getting extinct, how Tasmanian devils are getting extinct, and then used it to actually restore Californian condors whose numbers had dwindled from to 40 to increase it to 400. So this was enabled because, again, of these genomic technologies. There are very interesting questions you can ask. This is one of my favorite ones, that about a lice story. There are different kinds of lice. One is in the head, one feeds on fiber. But if it feeds on fiber, obligatorily fiber has to be there. So one very, very interesting study was done by a person. He asked a question: can it actually tell when did humans start wearing clothes? Because if fiber would not be there, then the lice would not be there. So he looked at head lice and body lice DNA all over the world, and he could actually find out when would the body lice have evolved from the head lice, and he could date it, because there are ways by which you can, when you see a change, at what time space this change would have happened, you can predict. So he could actually show that maybe 40,000 years back, this head body lice diverged from head lice. So that means people would have started wearing clothes during that time. And then there were excavation evidence of needles, which told that this could be actually possible. And he came out with a very interesting observation that maybe a large part of the human, during the evolution, we did not wear clothes, and we started wearing clothes 40,000 years back. So who would have thought that you can get a lice to tell this lice genome to tell this story? Other things are there. If you know variations today, then you can also predict why do you like a particular taste, why do you gravitate towards particular food. If you go to Subway, why do you want to prefer a particular kind of a sandwich? Because each of these food have different kinds of molecules in them, and our genes have different perception because of a variation to perceive sugar at a particular threshold, glutamate in the tomato at a particular threshold. So if we vary in these combinatorial places, we might want to eat different kinds of sandwiches even from the same Subway counter. So so why is this important? This information is important because if you knew that your taste threshold for sugar is a little high or a little low, then you can modulate your intake of sugar because of your knowledge about this genetic underpinning, and as a result of which would actually have a management of your health in a proper manner. So this is what is now called as nutrigenomics area, where people are looking at all these kinds of things for managing diet, lifestyle, etc. And the nearly the last part is that you can also kind of start to probe ancient system of medicine through the platform of genomics. So this is an area which I was deeply involved in the last 2 decades, where we again use the genomic technologies to be able to understand a basis or understanding of individuality of Ayurveda, upon which all the personalization happens about the diet, lifestyle, and environment. And we could show for the first time using these technologies that we could, if we do genomic studies on such kind of individuals, we could know why they are different at the molecular level. And these differences are also predictive of their different attributes, we could also discover that some Prakriti type adapt well to high altitude and some do not do well, even from sea level, looking at sea level. So that tells that if you were to marry Ayurveda based individuality understanding with genomic methods, you might be able to get a genetic basis to individuality, and that could be useful in tomorrow's health, wellness settings. So, this is the science of Ayurgenomics. I'm not getting into the detail, nevertheless, to show that we've been able to do lots of things once we had this Ayurgenomics framework developed in the country. And as a result of which, today we can think about how we can get the modern medicine and the Ayurveda doctors who look oppositely to be able to cross talk with this language of genomics. People now, when they know genomes, they know properties of microbes, they can synthesize genomes. So they can make genomes and which can have unique properties. And this is something called synthetic biology. So for example, vanilla is very, very, you cannot, uh, meaning for biodiversity also you need to not kind of kill trees, cull trees and all. But if you know what is the property and you can actually get it to the lab and try to get the microbes to manufacture them, something which is now being called as biomanufacturing. Such kind of properties you can get to the lab, make them in bulk, then you can also have a sustainable way of making products which could be of medical use and relevant in all spheres. And the ultimate, now this is my, I think the last but one slide, is that as I told you, the entire genome is very, very, it houses a huge amount of information. So, for example, if you take 20 grams of DNA, it can store all the world's digital data. So, as we are understanding, every year, every few years, whatever storage system we are having, it gets obsolete. And you cannot also use that into any other machine, right? But the DNA is so universal, it goes into every organism. So, can you actually think about storing information in DNA? And this is what is the latest technology has happened, that you can actually convert the byte information to a DNA information, and you can synthesize the DNA and put it in a tube. And when you want to retrieve the information, you just sequence it. So there's this interesting Netflix series called Biohackers. They have demonstrated this by putting their entire series into DNA. So they could sequence it and get back the entire information. So all the video, music, everything now can be converted into DNA. So this is the next generation storage which is being thought. And once you know that you can store it, then you can embed it. And if you can embed it, then you can embed this information into any device. So now people are talking about DNA of things, where you can embed an information into a material, which is like a bioprinting possibility. And so you have beads which are embedded, beads carry the information embedded, and that goes into a material which can be bioprinted, and then you can just cut it open, sequence this, and then you again have the information. So this, people demonstrated it by print a rabbit, they embedded it, and then they put this information in bioprinter, got the information out, again could make it print a rabbit, and it was reproducible. So you can pass on the information through the DNA in embedded system. So you can use this into, for if you want to carry information in any embedded material, then this is again a way of storage. And the final is CRISPR. You, once you get to know DNA, you get to know the property, you can change it for good, and then you can have for medicine, for biofuels, for creating disease resistant crops, for many, many things these can be useful. But of course, everything comes with a cautionary note. Whatever good you can do, there is a ethical side to it, and there could be misuse of it. So as these are growing, there are legal frameworks are also getting understood, and more people are trying to trying to kind of put rules in place so that misuse does not overrule the beneficial effects of these kinds of technologies. So finally, as I could maybe convey in this small lecture, that any environment, any ecological niche, or any pathogen load or culture can lead to adaptations, which would be the ones which get selected, or they are better off. It would be imprinted in the genome, and as these genomes move and migrate and mix with all existing genomes, they can actually create diversity, not only at the organism level, but at the organismal level. And these could be relevant or evident, all these events could be evident as patterns in the genome, which tells us about the uniqueness of biological systems, how their phenotypes have evolved, or the cause of diseases. And these could address questions at different sphere: where we were, where we are, where do we go from history, ancestry, culture, to impact of climate change, conservation strategies, human origin, how individuality comes, how to apply it in forensics, in diagnostics, in creating biologically inspired, for example, digital data storage solutions in genome editing, as well as in area like genomics. I'm sure many more would follow. So thank you all for patient hearing, and I'm there for questions unless I've kind of run out of time.
Yeah.
Thank.
Dr. Mitali, thank you very much. This was very, very interesting and useful. And you also brought in various Netflix series and others where people can see how people are applying some of these things to stories and movies and others. So thank you very much. We do have some questions coming up. I'll take a couple just given the time. One is, I think right at the end, you mentioned that this technology can be used for good and for bad, right? Is there a danger that just like every other technology that's been developed, people can misuse it? And what is being put in place to ensure that it is not being misused?
Yeah, so there are lots of legal frameworks which are coming on. Most importantly, as it is a small DNA sequence, it can be amplified, it can be put in any place. People, if they sequence it, they would not know whether it has come from a real person or a fake person. Just like you think about security concerns across everywhere, your DNA is very private. And if anybody misuses that private information, you could be at a risk. So if there are companies like 23andMe, Ancestry, they do, but when you get to know the ancestry, you can also get to know about ancestry of a person whom you would not want to be associated with, right? So you did not give the company the right to divulge that information to any federal agency, to misuse it. So many, as genomics is unfolding and people have started using it in various ways, many legalities are coming into place to be able to counter these. So this is a very dynamic field, and there are huge number of papers around this. It's a whole new system.
Thank you. Dr. Vitali, I wanted to just one question on a large number of companies around the world are getting funded and are started and are becoming successful based on creating medicines and various other things based on genomics. Obviously, we've heard of many large companies in the US, Korea, China, etc. Where does India stand on we have a great startup culture. Are you seeing a lot of startups or big companies doing, using this to create medicines, to create new plants or in many other fields?
Yes. So in the plant area of resilient agriculture, lots of things are happening. How you find through the genome sequence better breeds, ones which resist pests, so that you can introgress and make better varieties. So all those things are happening. In the human, we just got started, I would say, because we did not participate in the Human Genome Project, but we started when we were getting prepared. When the human genome comes, how do we start to analyze and understand it? Plus we have got a large, our populations are very diverse, very endogamous, unlike European populations. So we needed to know how we are as a population, so that anything we do, it would be useful for the diversity of population. So large number of efforts have happened in the rare disease space, where government has put in Nidan Kendras and testing centers so that we know which population is burdened with what kind of diseases, so that targeted interventions are possible, prenatal screening, etc. And there are a large number of setups. So Genome India project, which has sequencing 10,000 individuals, it's a project from DBT, is actually looking at populations all across the country. In terms of medicine, there are now people have started using it for reducing the time to drug discovery, because this is also got initiated. Cancer genomes, people are looking at how to understand or manage stages of different kinds of cancer. So initiatives like ICGA has happened. So developments are happening, and at this time, in terms of technologies, we are quite at par, but in terms of scale, we are not at par, and we need to do much, much more. And since this is a diverse population, we need to contextualize it, which is what we cannot just borrow everything from the West. We have to develop our own indigenous know-hows. So
That's yep. Dr. Vitali, we have run out of time. This was very, very helpful. A great journey into understanding what is genomics, where it came from, where it's going, and why we need to understand it. So I thank you on behalf of Mission Karmayogi. I thank you for joining us today. I'm sure this will be viewed many times, and many people might reach out to you after this. Can you just, as we end this, can you suggest a couple of books that people should read if they want to know more about it, but please don't give us textbooks.
no, there are lots of popular books. One is Genome in 23 chapters, from where one can begin. Then one can read Survival of the Sickest. There's a book called Age of Scientific Wellness by Leroy Hood. Then there are lots of books on genomes. Uh, One book called by Jared Diamond, Guns, Germs, and Steel, is very, very interesting, where people can get an idea as to why genomics could be important. Of course, that doesn't talk about genomics any bit. Then there are books on culinary genomics, eating or cooking the food in the right way. That's also area. Gut, I had shown you the book called Gut. Then there's a book on personal genomes. So if you, I have written a book by World Wildlife Foundation with a small kid called Genome Jaunts. It's for the kids to know curious animals and how they come about. So it was written by an eighth standard kid and a PhD student. It's called Genome Jaunts. World Wildlife Foundation has printed it. I do not know whether they are still printing it. And many more.
That's quite a list of books. I think I know that you will have suggested many, but I think this is a great list. Once again, Dr. Mitali, thank you very much for joining us today.
Thanks.
Thank
Thanks.
you.
Thanks.